talking specifically in our biology class on the cell. So what's a cell basically? Now, early scientists like Robert Hooke, they came up with theories about the cell, and from these cell theories we are going to list the 5 cell theories, and then we are going to define the cell from the cell theories. The first cell theory was that cell is a basic structural and functional unit of life, and that is the definition for cell. So you can simply define a cell as the basic structural and functional unit of all living organisms. The second cell theory is that all living organisms consist of cells. That means all living organisms are made up of cells. So that's the second cell theory. And then the third cell theory is that there is no life apart from the life of cell. That shows that when you see cell, then there's probably life, but anywhere there's no cell, then there's no life. The other theory is proposed by scientists, and I will list 5 of these scientists for us. And those 5 are Robert Hooke, as I mentioned earlier, and he was one of the earliest to propose theories in biology. And in 1665 he proposed. He was one of the scientists who proposed cell theory. Also, we have Felix Dujardin in 1835. We also have Matthias Sledan in 1838. We have GlEDUzownë in 1839. We also have Rudolf van Vichoid in 1855. So you can see that these scientists, scientists and biologists or naturalists, they lived during the 17th century and 19th century, and most of their works, they are still recognized today. So basically today, before we delve into the cell theory, we've defined a cell, and basically there are different structures of a cell. Before we go into the structure of the cell, we are going to talk about the various equipment used to study cells. And as you know, cells are very tiny, very tiny. They cannot be seen by the natural eye. So most of the cells, they are usually observed and experimented on by means of equipment. And one of the major equipment used to recognize and study our cells is called a microscope. And basically a microscope looks something like this. You have something like this. You have a part that goes this way. Then you have a part that holds it like this. Then after this, you have what is called the stage-like part. Then the base. You have a knob. You have a stage. You have So this is like a short, a simple version of what a microscope is, but it contains the basic parts of the microscope, and then we also have something below. So one of the first basic parts of a microscope is your eyepiece, and then your eyepiece is these 2 structures that extend at the top of the microscope. This is called your eyepiece. And then inside your eyepiece, this is where you look into. You look into your eyepiece when you want to look into something microscopic. And when something is microscopic, is beyond the size of the normal human eye, because light has a specific wavelength. If you are a physics student, you would have heard of wavelength. And wavelength is like the how much of light can penetrate something so that we can see it. So, but the wavelength order of cells are very much smaller than those of light. So we have to use much more smaller equipment, or much more detailed equipment. So this is called a simple microscope. And then these are the eyepiece. Then inside this eyepiece, there's what's called a binocular lens. And that binocular lens, it is the lens that is a convex lens that magnifies the image by several times, sometimes thousands of times, so that you can see in detail what we are looking at. Then we also have, this is the support of the, this is the base of the microscope. Then this is the coarse adjustment knob, COARSA. So this coarse adjustment knob, it helps us to adjust the object and the light falling on it so that we can see it more clearer. And then this is called the condenser. And this condenser is just like a part of the microscope that helps us to bring lights together. Just like a converging lens, it's more like a mirror. It brings the light and then converging it on the object. And this is where you put the object. The object is put on the stage. So this is kind of like a part where the light passes through and then the object can be seen on the stage. So the object is often placed on a slide. So and a slide is basically a flat it could be a glass or plastic and transparent substance, part that allows light to pass through it, and where you can put on which you can put your samples, and those samples contain what you want to see beyond your eye. So now this is called your slide. Then after the slide, we also have the this is another lens. This is called the there's the objective lens. This is the objective lens. Yes, this is kind of like the objective lens. So and this objective lens help us to see the object in detail. So they are often closer to the object, and they often have they are often like more. They are often stronger than the eyepiece lens. So there are two different types of lens in a microscope. There's your objective lens and eyepiece lens. So this is just a simple demonstration of how, or illustration of how your microscope works. So and this instruments like this, which were developed in the early days, were used by scientists to help us understand these structures better, cells. So examples of Leeuwenhoek was an early scientist who developed a simple microscope by which he was able to check the components of many substances, and most of them discovered that they are consisting of cells. So these cells, there are different types of cells. But before we talk about the different types of cells, let's talk about the form where we can see cells. We can see cells as existing alone, and by now you have known that if you know that the body consists of cells, if you know the body consists of cells, you should know that those cells will be numerous in the body. So like our skin has, for example, our skin has epidermal cells. So these cells, they have different names based on their function and structure. That is why we say the cell is the basic structural and functional unit of life, right? So now these cells, for example, some of them exist alone, like like an amoeba. Amoeba is a cell, and whenever they exist alone, they are called unicellular organisms. Then some of them, they exist in filaments. So and those filaments is basically when the cells are joined from end to end. So a filament looks like this. You have the cell, and then the another cell is joined to it at the end, and that to 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 the end. Example of this is spirogyra. So it is kind of like that green substance you find in all these dirty places. So and that is a unicellular organism. It's specifically kind of like a thylophyte. So and we'll talk about those thylophytes later on, which I've kind of talked about before in a previous video of this channel. So thylophytes are special parts and types of plants. So that apart, lastly we also have cells existing as colonies. So example of colonies is volvo, and what colonies means is that those cells just come together like a crowd, a mass of cells, just like when people gather in a building. So those cells, they gather together, and often they could be in any shapes. For example, if you have a colony of volvo, you basically have different cells in that colony. So these are examples of colonies. So the cells are joined together, not end to end, but in mass. That is what a colony is. So basically, I've talked about the three major forms of cell, which is they can exist in unicellular form, where they exist alone. They can also exist as filaments when they are joined end to end, and they can also exist as colonies where they are massed together like a ball of cells, like a mass of cells, and that mass of cells can have different shapes. For example, the cells of the eye, many of them are like a mass of cells. But then there's what we call differentiation. So and that differentiation, I will just explain it simply, but that's not my aim. I don't aim to explain it in this video, but let me just give you a little snippet into what it means. Now, a differentiation of cells is basically when those cells have the same function, just like twins. You know, when you have twins, when you see twins, they are alike, right? So and they share different things in common, both physically and anatomically and physiologically. That's in their body. So basically, those twins, they are just like those cells. Those cells are like the twins, and when they are plenty, they all act in the same way, and they're like synchronized. So and what that means is that those cells, they have a specific function, and when they have a specific function and the same location in an organism, we call it differentiation. And that cell differentiation occurs in special organs of the body, like the eyes. So once cells are differentiated, they can form organs, and they can form other parts of the body. So that is just what I'm trying to explain. But moving on, after the forms of cells, they can exist in singular, they can exist in colonies, they can exist in filaments. We also have the fact that there are different types of cells. Now in our today world, we can distinguish between two fundamental types of living organisms. Fundamentally, we know they are plants, and fundamentally, we also know that they are animals, right? Now, those plants, they have their different types of cells, and those animals have their different types of cells. For example, when we talk about plant cells usually have a definite shape. If you ask the difference between plant cells and animal cells, plant cells have definite shape, while animal cells do not have definite shape, right? So now, plant cells often look rectangular, something like this, and plant cells have cell walls. So at that cell wall, animal cells do not have the cell wall. So and then you have the nucleus. The nucleus is often and even in plant cells, your nucleus is located at the side, where you have a structure known as the vacuole. And now in every cell, even in our body, we have a structure in our body called the kidneys, which help us to pass out unnecessary parts of our body, unnecessary substances. So that vacuole in the plant, it helps the plant to pass out unnecessary substances, just like water and chemicals that are not needed. So and then we have the cell members, and these are often known as plastids. So in plants, we have different substructures of a cell. In the cell, there are different structures that are beneath the cell. In the ancient Greek and Roman philosophy and science, they often believe that the cell is the simplest part of a substance. But nowadays, we know that cell consists of smaller particles, and those particles, some of them are plastids and some of them are nuclei. So and those nuclei, they are often dense, and that nucleus of a cell, you've often heard of the word nucleus, right? When you talk about the nucleus of a cell, the nucleus of a cell is like the base of the cell, like the father of the cell, it's more like the most important part of the cell. Though let's say it is one of the most important part of the cell, it's not the most important. So and this nucleus, along with the vacuole, along with the plastids, along with other different parts of the cell, make the cell to work together in harmony. And the outermost part of the cell is known as the cell wall, and this is a plant cell specifically, cell wall. Well, I must add that animal cells do not have cell wall, only plant cells have cell wall. Then the innermost boundary is called the cell membrane. Now, in both animal cells and plant cells have cell membranes, but animal cells do not have cell wall. So basically, this is a typical plant cell in simplified form. So this is, though if you are asking your exam, you might be asked to add more structures. Like on average, you might be asked to label 8 parts. So and then we have what's called the mitochondria. And then the mitochondria, basically what it does is that it helps the cell to break down food. Mitochondria is just like the intestine of the cell. Now, if we can also reason by abstraction, because most of what we see in our today world, like even in our body is like an abstraction of the cell. Basically, if you think of our body as a cell, every cell has different parts of this body. So that shows that a cell can even work on its own or work in conjunction with other cells. So and even when the cell is working on its own, it has its own intestines. Its intestines are the vacuole, the food vacuoles, and then the lysosome. The lysosome is a part of the cell that helps it to break down food. The mitochondria is a part of the cell that helps it to break down, to digest. So it basically respirates the food taken in by the cell. So I know that, and now you know that there are different functions of organisms. Without being told, you know that even from your basics, I assume you should have heard of Mr. Ninja, which is a mnemonic for remembering the fact that there are a lot of functions of cells, which are movement, respiration, nutrition, irritability or sensitivity, growth, excretion, respiration, oil production, competition, adaptation, and death. So living organisms, they live and they die. So those functions of organisms, those properties of organisms, they are always ensconced in the structure of the cell. So basically, this cell, every structure you see in this cell There are many more structures. Even scientists have not known all the structures in these cells, because we keep discovering some, and some are still new, some are still developing. So now this cell, and even when cells adapt, they kind of change some structures. And even when we have some like genetical engineering, where we engineer some part of the cells, so these cells, we can make these structures function differently. So we'll talk about that more in subsequent videos. But basically, these structures, they have, each of them has a function in from Mr. Nejadi. So for example, this nucleus can help the cell to reproduce, because the nucleus contains the chromosomes and the genes of the cell. And by now you should have known that a gene, when we talk about gene, is like the part of the organism that transmits its basic nature. Like when we involve reproduction, we also involve transferring of genes and alleles, and then chromosomes and other genetic material. So basically, then animal cells are just like plant cells, but they don't have form. Animal cells are often formless, just like an amoeba. The cell of an animal of an amoeba has is a partition like this. These dotted lines are a partition between the exoplasm and the endoplasm. So the endoplasm is within the partition, while the exoplasm is beyond the black partition. Then we also have the nucleus of the cell, but the nucleus of an animal cell is often located in the center of the cell. Then we also have different other parts of the cell. We can just represent them by dots. But if you are writing your exam, you should be able to go in detail and name the different basic parts of the cell. So basically, in conclusion, we've talked about the cell today. We've talked about the cell history, how different scientists like Robert Hooke, Felix Dujardin, Matthias Schneider, GlEDUzownë, and Rudolf van Beechoid in the 18th and 17th century or 19th century, how they've contributed to our basic modern idea and representation of the cell. And then I've talked about how we can abstract the function of an organism from the function of the cell, that the cell can exist independently. It can exist as a filament where they are joined from end to end. It can also exist as a colony where they are joined like a mass or ball of cells. We've also talked about an instrument used to study cells, and that's the microscope. We talk about the eyepiece, the objective lens, the base of the microscope, the coarse adjustments, and all the other parts of the adjustments. And basically, you can just see it as a combination of structures and lenses that help you to magnify the object you are trying to look into. So basically, that is all for today's video on